Okay, so we talked about southern blot. The key in southern blot is to start with genomic DNA or any DNA. You start with DNA, you digest it with a restriction enzyme, and then you throw it. There is another blotting system called Northern blot. Now this was not discovered by somebody named Northern. It was simply because they took southern, so they converted the name to Northern. Just scientists trying to be funny. And basically, it's a blot where you transfer, instead of transferring DNA onto a membrane, you're transferring messenger RNA onto it. Right? So what's the key difference between DNA isolated from a cell and messenger RNA isolated from a cell? What is the messenger RNA when you isolate it from a cell telling you? What's it telling you about genes and things of that nature? When you isolate messenger RNA from a particular cell, let's say skin cells. You isolate DNA from skin cells. What's the DNA telling you about the DNA from skin cells? Throw something at me.
Well, the DNA is just the whole recipe book.
Correct. The DNA is the whole recipe, that it's your entire genome, correct?
Yeah.
Okay. Now what happens if you isolate messenger RNA from a skin cell?
If you isolate messenger RNA, it reveals what genes are actually being used.
Correct. So when you isolate messenger RNA from a skin cell, it's telling you the genes that are active in that cell. You're only getting a picture of the genes that are active in that cell. So Northern blots is an analysis of genes that are active in a particular cell type. That's what it's telling you. Okay. And so you do the experiment more or less the same, not exactly the same, but more or less the same. This is a rapid method that allows you to determine the cell type in which a specific gene is active and being transcribed. Okay, so let's imagine there is an embryo. The embryo is going to differentiate and become an adult, and it's going to produce different cell types: brain, bone, blood, lung, liver. Okay, these tissues differ between each other because they're transcribing a unique subset of genes. Each tissue has a unique subset of genes. Some genes are of course common, but there are a very significant number of genes that are only made in brain cells or bone cells or blood cells, etc. So each tissue contains a unique and distinct messenger RNA population. So the presence of the RNA in this mixture is a reflection of gene variation. So let's imagine Northern blots basically are like southern blots. But instead of DNA as the starting material, you're using RNA, messenger RNA. You take the cells, you break them open, you isolate the RNA. And now the difference is, remember, restriction enzymes do not digest RNA. So for a Northern blot, you don't digest the RNA. You take the RNA and you run it directly on the gene. And you transfer the RNA to a membrane just like before, and you use a radioactive probe for that one gene that you are interested in. So the RNA can be isolated from different tissues. So let's imagine you have an individual one. You isolate RNA from different tissues like lymphocytes, erythrocytes, brain, bone marrow, and kidney. And you use a specific probe, let's say the globin gene. In this individual, when you use this RNA, you run the RNA out, the RNA will look like a large smear. You probe it with the globin probe, it will light up in erythrocyte, RNA isolated from erythrocytes, and RNA isolated from bone marrow. The globin gene is not active in lymphocytes. It's not active in brain. It's not active in kidneys. And so you will say, the globin gene is active in red blood cells and in cells of the bone marrow. Now in individual 2, you do the same experiment and you find the bands don't light up. most likely individual 2 is dead, but for this example, it just tells you that the globin gene is not being made in these cells, in this individual. And so that's a reflection of activity. So you get a picture. When you see a band, it indicates that gene is being made in RNA in that cell type or in that individual. The gene is. So there is another, so this is known as a Northern blot. The key thing to remember is Northern blots, you're analyzing RNA. In Southern blots, you're analyzing DNA that has been digested with a restriction enzyme. That's the difference between the three blotting systems. Otherwise, it's exactly the repeated model system. Okay, so this is great. I can now analyze RNA from different cell types. And I can analyze it for one gene. Let's say the globin gene. And maybe tomorrow I'm interested in another gene. I will, let's say the histone gene. I will do the same analysis, isolate RNA, or take the same RNA from these individuals and probe it with a probe for the histone genes. Or another gene that's interesting to you, it's up to you. So you can do individually, you can run one probe after another probe after a third probe, one after the other. Set up a system. Every day you run one probe. After 30,000 days, you will have a picture of all 30,000 genes in the human genome. And you'll be able to say, aha, now I know after 30,000 days, or however many years that is, you will get a picture of the entire human genome with regards to which genes are active in which cell types. So you can use Northern blots to do that. It's extremely, extremely slow and time consuming because you can analyze one gene at a time with one filter. There is a new method that was developed in the 1990s called microarrays. And that allows you to get a picture of the entire genome, all 30,000 genes, in one single snapshot. So, what are microarrays? Microarrays are used once you know the sequence of the entire genome of that particular individual or species. So imagine here is an individual who's got this piece of DNA. Each fragment of DNA has a particular sequence. Here is gene 1, it's got the sequence AAAAAA. Here is gene 2, it's got the sequence GAGAGA. Gene 3 has the sequence GGGGGG, and gene 4 has the sequence TATATA. So, you could do this experiment, you could do a Northern blot, and you would probe it with 4 different probes on 4 different days, for example. On day 1, you would probe it with a probe that's specific for gene 1 and analyze where it's being made. Next day, you would do it for gene 2, the third day for gene 3, the fourth day for gene 4. You could analyze it in the gene. Or, you could use a microarray, and you could analyze all 4 genes at the same time, all together. So, the key is, take a glass slide, literally, no kidding, it's a literal glass slide, like the little microscope slides that you have all up in the neighborhood. And you on tiny, tiny pinprick, you stick a probe that is made chemically that is specific for gene 1. So on the slide, you will stick a piece of DNA, single-stranded DNA, that goes AAAAAA at this point on the slide. Next to it, you will put another piece of DNA that goes GAGAGA. Again, very specific sequence. This sequence is only found with gene 2, not 1, 3, or 4. And then a totally different slide, you would stick the sequence GGGGGG. This is found for gene 3, but not for 1, 2, and 4, and so on. And so in effect, on a single glass slide, you can put 30,000 pieces of DNA. Each one is specific for a specific gene, for example. So now, you have all 30,000 genes arrayed in little dots with the DNA sitting out there, single-stranded DNA sitting out there. waiting for something to happen. And each dot, we know exactly which gene it corresponds to. So we know dot 1 on the slide corresponds to gene 1, dot 3 on the slide corresponds to gene 3. Right? And dot 6 over here corresponds to gene 6, and so on. And so on. So we know where exactly each gene is located on this little slide. Now, what did we do in the northern blot? We basically transferred the messenger RNA onto a filter and probed it with a radioactive globin probe, correct? The messenger RNA was not labeled. The little globin gene was labeled, correct? Yes? Clear so far, right? That's what we've done here. This globin probe was labeled. The total messenger RNA was unlabeled. With the microarrays, you're going to flip the whole system over. You have all of the probes stuck on the slide and they are not labeled. So these little fragments, these are your probes. This is probe 1, probe 2, probe 3, probe 4. These are not labeled, they are stuck on the slide. You isolate the messenger RNA from a cell and you label all of the messenger RNA with radioactivity or a fluorescent dye, usually a fluorescent dye. So it's going to give out a signal, a fluorescent light signal. So you're going to isolate RNA or whatever from a cell. You're going to label it with a fluorescent dye. And now you're going to take this labeled RNA and you're going to anneal it onto the microarray. And it's not really RNA that's labeled, it's the cDNA that's labeled. So you made a complementary copy of the RNA, and that complementary copy of the RNA is what is labeled. But it doesn't matter. Assume that all of the RNA is labeled. This RNA is now added to the slide, and it will hybridize. So here is the slide. You add this RNA into the slide. It will hybridize. Yes.
Do you know what the complementary strand is going to be, and how do you not know?
Excellent question. Excellent. So we don't know what the complementary strand is going to be. So how do I make a cDNA? Correct? So what's a messenger RNA? This was question 1 in the paper. What are the 3 characteristics of messenger RNA that are different in eukaryotes from prokaryotes? They have a cap, they undergo splicing, and what's the third important thing? Poly-A. Right? So, all messenger RNAs have a tail made up of atoms. So, when we make cDNA, and we want to make a copy of it, complementary copy, we start, so this is 5 prime to 3 prime. When we want to do this, we always start with a piece of DNA, 5 prime to 3 prime, called oligo DT. So it's got just a run of T's, usually 25 to 30 of them. It only hybridizes to the tail of all the messenger RNAs. Now when you add reverse transcriptase, it copies the messenger RNA, and it copies all of the messenger RNA. So you have no idea what they are, but you know you've amplified or you've labeled all of them.
And then you just
You use that entire mixture and throw it onto the slide.
and you just improve it at
That's all. So, you've labeled the RNA as complementary DNA, as you just saw. And you're going to add that onto the slide, and it's going to hybridize. Once again, complementary base pairing. So you can see how important complementary base pairing of double-stranded DNA is to all of the experiments we've been talking about. From restriction digestion to PCR to blotting to whatever. Every aspect of molecular biology depends on complementary base pairing. So So based on this base pair, you will get DNA fragments, cDNA fragment hybridizing to the little probes that you attach to the slide. And then you have a specific camera that takes a picture of all the fluorescent dots, and you can see little dots appear, fluorescent, and they light up, and you will say, ah, this little gene was active because it's hybridizing and it's giving off a fluorescent signal. This gene was active because it's hybridizing and giving off a signal. This gene was active, and so. So when you do this analysis, let's imagine here is an individual that's missing. So in this individual, you've got 4 dots indicating that gene 1, gene 2, gene 3, and gene 4 are being made into RNA. Because you've got 4 fluorescent dots on this slide. In a totally different individual, if I were to isolate RNA from here, or from this individual is missing 1 gene. This gene is not either being deleted or it's not being made into RNA or something. When you isolate and make cDNA, label cDNA, you're going to hybridize it to the same sort of microarray. All 4 genes are still present. This will anneal, this will anneal, and this will anneal. But this gene here will not be able to find its partner because there is no partner. So when you label it and you take a picture, you get 3 dots, and this dot will be blank. And that way you'll be able to tell that in this second individual, this gene is missing, and it's not being made into RNA. And so you'll be able to tell.
Why is the complementary RNA,
Because it's
why have you written T instead of G?
because it's cDNA. Remember, you take a messenger RNA, you're going to make a copy of it with reverse transcriptase. You're converting the copy of the RNA into DNA, right? So, basically, a microarray is a reverse northern blot. In a northern blot, you had RNA on a filter, and you were probing it with a probe for a specific gene. On a microarray, all of the gene probes are on a filter or a slide, and you're probing them with the RNA, in a sense. It's not really RNA, it's a complementary copy of the RNA, but it is RNA in a sense because you can work your way backwards. And so that's what it is. So it's a reverse northern blot. The beauty of it is you get a picture for all 30,000 genes on day 1. You don't have to wait for 30,000 days if you're doing 1 gene probe a day. So it's a very efficient way to determine the expression of all 30,000 genes in a particular cell at a particular time. So you get a genome-wide view of this whole story. It allows us to examine gene expression of all of the genes in the gene. Each spot is DNA for one defined segment of the DNA. It's spotted on the grid, and you cover the entire genome. So you have a slide, you have these dots. You make total RNA from individual 1 and individual 2. Let's imagine. So you're making RNA from one individual and another individual. You're going to label the total RNA differently. Individual 1 will be labeled with, say, a red dye, individual 2 with a green dye. Right? And then you're going to add those onto the dots, onto the microarray. Add the labeled RNA and let it hybridize. And then you measure the amount of fluorescent signal that comes off. And here is the result. Let's focus on just this one row of dots of genes, 5 different genes. In individual 1, gene 1 is active because you're getting a nice red signal. Gene 2 is active. Gene 3 is active. The genes 4 and 5 are active. That's in individual 1. In individual 2, gene 1 is active. Gene 2 is probably more active. Gene 3 and gene 4 are not active, and gene 5 is active. And so you can see how this whole system works. You can determine the ratios of individual 1 for gene 1 to individual 2. For gene 2, you can follow the ratios, etc. can reflect the answer and the results as ratios. You can say there are 5 molecules of gene 1 being made in individual 1, 5 molecules for gene 2. But for gene 2, you have 5 molecules for individual 1, but you have 10 molecules for individual 2. So the gene is more active in individual 2 and less active in individual 1. Or vice versa. It's less active in individual 1 and more active in individual 2. Whatever. So you can see how you can measure these signals, and from that you can say, you can talk about all 30,000 genes for each individual. You get a picture of the entire genome and its expression pattern. And you can do this for one cell type, all cell types, it's up to you. How many times you do it. There are many, many different types of cells in the human body. You can isolate RNA from all of those cells and individually analyze them by microarrays. But you can also use microarrays for other things. It's not just RNA and DNA that you analyze by this method. You can use microarrays to actually analyze gene deletions. To measure changes in transcription is obviously one important method we use for microarrays. But you can also identify gene deletions. And the way you do that is simply you isolate total genomic DNA from different individuals. You label the genomic DNA and use that as a code. If a gene is deleted, it won't hybridize to the microarray at that one particular spot. And so you'll be able to determine which gene is missing from one individual but present in the second individual. And so microarrays are used all the time now, more often than not, to analyze deletions or amplifications of entire segments of genomes. If you have more copies of the genome, you can analyze that. Okay, so with that, this...
Question?
Yes,
How long does it take for this hybrid
overnight.
overnight?
And then the next day you have a result. So 2 days at the most, if you're a little slow to do this. Obviously you have to get it to work over and over. But these days there are companies that do this for you. You send them the RNA, they will do the analysis for you. They'll do all that contract labels. They just keep, and so they've become so good at it that it's very easy to get information. But the problem is really analyzing the information. When you get back information for 10 different individuals or 10 different tissues, 30,000 genes or whatever, it's all that terabytes of data that you have to sit and analyze. That becomes the rate limiting step is the analysis, not the generation of the data. Okay, so we've got time, so I'm going to start with the next lecture, but we'll probably we'll go through it one more time again. So we're going to now move more and more into studying individuals and studying the similarities and differences in individuals. So we're now going into what's known as personal genomics. This is the work that's been going on for the last 10 years, since the human genome was sequenced in 2001, 2002. In that area, I can't remember. No, 1999, somewhere, I can't. One of them. But it's basically, we're now into the 21st century. So we finally reached this point, and we're going to analyze similarities and differences in individuals. And so now here we're basically talking primarily about humans. We're very rarely going to talk about similarities and differences between individuals in different species, though you can use the exact same method to study individuals in different species. And there are 3 different areas we're going to touch on. We have little acronyms: RFLP, SNPs, and repeats. Okay, so RFLP stands for Restriction Fragment Length Polymorphism. So restriction fragment lengths, you know restriction fragments have particular sizes, and polymorphisms, fancy way of saying differences in size. Okay? So you're going to analyze the differences in size of a particular restriction fragment between different individuals. SNPs, single nucleotide polymorphisms, or point mutations, if you wish. And then repeats are the third aspect. And we're going to start actually with repeat, or no, maybe not. Okay, so I'm going to start with, yes, we're going to start with repeats first. So We're going to go backwards. We're going to start with repeats and then we're going to go to RFLP and SNPs. So imagine there are three different individuals right here, and they have a particular sequence in their genome. Here it is. And there is a G over here at the fourth nucleotide. And then there is a repeat. GC, 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 GC. That's a repeat, correct? Two nucleotides being repeated many times. Individual 2, the sequence looks more or less the same, except this G is converted to a C. And the number of repeats are different. So in this individual, individual 1 has 1, 2, 3, 4, 5 repeats of GC. Individual 2 only has 3 repeats of GC. Individual 3 has 1, 2, 3, 4, 5, and 6 repeats of GC, but has no change at the G. The G is still the same. So, this change of a single nucleotide is called a SNP, single nucleotide polymorphism. Change of one nucleotide. This number of repeats changing is just repeat alteration or repeat polymorphism. So the number of repeats changes, but the repeats themselves don't mutate. They're still GC, GC, but they're not repeated as many times as the same in all individuals. There are different numbers of repeats in different individuals. So, genetic polymorphisms, and here we're not going to talk about mutations per se, we're going to talk about polymorphisms. But mutations are a subfraction of polymorphisms. So genetic polymorphism is a difference in DNA sequence amongst individuals, or amongst groups or populations. So, and they come in two different primary categories. You get single nucleotide polymorphisms, what we've been referring to as point mutations until this point, and then repeat heterogeneity, or number of repeats being different in different populations. So, two different categories that we're going to focus on. So repeats are a way of, the number of repeats at a particular locus is a pattern that is specific for a specific individual, and it's referred to as DNA fingerprints. So if you've heard of DNA fingerprinting, what we're talking about are the repeats at a particular locus. And that signature is your particular DNA signature. Okay, so there are sequences in the genome called satellite sequences. These are short sequences that are repeated many, many, many times in the genome. And you can imagine these repeats being interspersed, i.e. they are spread out throughout the genome. Imagine there is an individual with 2 chromosomes, and this sequence, a particular sequence, whatever that sequence is, A, or GC, 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 GC. That sequence may be present here. It may be present totally different position, but it's the same sequence repeated. Or it could be present on a different chromosome. These are known as interspersed repeats. They are dispersed throughout the gene. Alternatively, you can have the repeats and they're all in tandem, one after the other, like I just showed you, GC, 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 one after the other. And that's known as a tandem repeat. It's present at one particular spot in the genome, and there are many copies of it. So there are two different types of repeats, interspersed repeats and tandem repeats. Okay, yes, question?
How many days, like, technically you could just went out and you have many repeats, so like a minimum of 2 repeats.
So we come to that. Depends on what you're studying. If you're studying interspersed repeats, you usually use larger fragments, like 10 or 15 nucleotides. If you're using tandem repeats, then 2 is more than sufficient. Okay, so there are different types of repeats. There are satellite repeats. These are very large repeats. One repeat could be 100 nucleotides long. That's repeated again, and repeated again, and repeated again. Or it could be up to 1,000 base pairs long. It could be repeated again, and repeated again, and repeated again. Regions at your centromeres and your telomeres consist of these repeats. They're large repeats that are repeated many, many, many times. So yeah, the centromere in the middle of the chromosome, the ends of the chromosome, telomeres, they basically consist of these repeats. Microsatellites are extremely small units. These are usually between 2 and 5 base pairs. Like AGC, 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 or GC, 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 GC. So that's where they come in. So those are known as microsatellites. And mini satellites are slightly larger. They're between 6 and 100 base pairs. So micro, mini, and then the nano. The 3 different grades of routines, basically. Okay. Now these routines